working in the environmental and natural sciences field, an opportunity to share their work with peers, policymakers, and the general public. Currently, due to the effects of the ongoing global pandemic known as COVID-19, we have adapted the conference to the situation and are now in our 4th week of our BNHC virtual webinar series, shortened to BNHC-V. This week's webinar session will be focusing on one of the most amazing groups of animals, the reptiles. So last month, if anybody had participated in last month's wildlife webinar sessions, I focused on Bahamian wildlife and invasive species such as cane toads. Today, we are going to be focusing more on invasive reptiles as well as on sea turtle work done in the Bahamas. So our first presenter is Annabelle Brooks, who is an adjunct scientist at Cape Eleuthera Institute and a PhD candidate at the University of Exeter. The title of her presentation is The Description and Prevalence of Fibropapillomatosis. I totally messed that up, okay. Disease in green sea turtles throughout the Bahamas. So without further ado, we're going to bring her onto screen. Okay.
All right. Thank you, Scott. Good attempt there at the pronunciation. This is the fibropapillomatosis disease. So yeah, good morning, everybody. Thank you for joining. Thank you, BNT, for inviting me to speak as well. So this talk is going to focus on the description and prevalence of the fibropapillomatosis disease in green sea turtles throughout the Bahamas. And I just want to acknowledge all the co-authors on this study. So first of all, we have Dr. Karen Bjorndal and Dr. Alan Bolton from the Archicast Center for Sea Turtle Research at the University of Florida. And we have Stephen Cannett and Barbara Crouchy from Family Island Research and Education. Dr. Beth Whitman, who is at Florida International University. Dr. Mariana Fuentes from Florida State University. And then also the Geronimo program, which is a sailing at sea semester program that travels throughout the Bahamas that Stevie Cannett actually started. So all these various groups have been really integral to be able to make this study possible. So I want to thank them and acknowledge them as well. So I'm going to focus first of all on this actual study and then just go on to talk about some of the other kind of wider threats that are still facing sea turtles here in the Bahamas as well. So first slide here. Oops, there we go. Okay, so hopefully many of you have actually had a chance to see a green sea turtle. It's by far the most common turtle that you would expect to encounter here in the Bahamas, mainly because it spends its time in coastal habitats. So you might be walking along the shore, or if you're spending time in a tidal creek, in a mangrove area, you're more likely to see this sea turtle than any of the others. And this is because after it spent its first few years of its life out in the open ocean, developing from a hatchling, it eventually settles out into these coastal habitats to go for its developmental stage, for its juvenile period up until sub-adulthood. And at that point, they'll be large enough to then head out back into deeper waters again. And the kind of habitats, like I just mentioned, are these mangrove creeks. So the Bahamas is covered in these amazing habitats. So the tidal seawater is flowing in and out over the course of the day, making these really nice shallow areas where sea turtles can avoid predators like larger sharks and in this structure, and also providing food for them as well. So the green sea turtle is mainly herbivorous. It actually starts off its life carnivorous. Out in the open ocean, it'll actually feed on small crustaceans and larvae and things like that. But when it heads into the near shore, it becomes herbivorous, feeding mainly on seagrass. And they eat a lot of it. So that's their main source of energy. And you can see here in this image on the left is the cropped area of seagrass, so where the turtle has repeatedly grazed in the area. And they tend to nibble off the longer piece of the seagrass, which floats away, and then they actually feed on the shoot there. And as they revisit the new shoot, it's more nutrient rich. And you can see on the right an area which hasn't been grazed. And this action is actually really important for that wider ecosystem. So by maintaining this kind of grazing, they keep the seagrass ecosystem healthy. Therefore, other organisms, for instance, such as conch and other juvenile fish that live in these areas, they're able to take advantage of this habitat. And if the grazing doesn't occur over time, the seagrass actually overgrows and shades itself and starts to decompose as well. So they're really important in these kind of seagrass and mangrove ecosystems. Also, they have a really important, big importance regionally as well. So in this figure, I want you to focus there where all those arrows are merging is actually in the southern Bahamas down in Great Inagua. And a genetic study that was conducted showed that the green sea turtles, the juveniles that spend their time here, actually originated from all these other countries where you can see the silhouettes of the turtles. So turtles that we see here in the Bahamas, we can expect they were actually born anywhere within the southeastern United States, further down the Caribbean, Central America, and even as far as South America or the west coast of Africa. And after their hatchling phase out in the open ocean, they eventually settle down here in the Bahamas to develop. And one day, those turtles will return to further reproduction and nesting as well. So it's really important that the turtles that we see in the Bahamas, that we understand that they are not our Bahamian turtles, but they're regional Caribbean turtles for the most part. And therefore what happens to them here in the Bahamas is also important for what happens there. And so it's of great importance as well that in 2009, the law was passed banning all harvest, possession, take of sea turtles and their products. And to be clear, they are an endangered species, which was the reason for this ban to be put in place, and they still currently are, which means that their populations have been quite heavily reduced over the last few centuries. And throughout the Caribbean, they're at less than 10% of what their numbers used to be pre-exploitation. Okay, so that was just a little bit about our green sea turtle, and I'm going to move on to talk a bit more about the actual study. So when I say the word emerging infectious diseases, if I ask you to think of, just on the top of your head, think of a disease that maybe you've been hearing a lot about, you're most likely going to be saying COVID-19, I'm guessing. And so this is in fact actually an example of a recently discovered disease. Maybe it's increased in incidence or geographic range and moved into new host populations. So that's what an EID is, just like COVID is. And the causes for this could be anything from introduction of domestic or wild animals into habitats, new habitats, maybe their movement of like trafficking of animal and plants, agriculture, and so on. Anything that increases like a host population, for instance, can cause this rapid spread of disease. Okay, and there's increasing evidence that these kind of diseases are linked to human-induced environmental degradation, both in terrestrial and marine systems. So I'm going to give you a few lists of those here. So, and when I say environmental degradation, I mean things like habitat loss or habitat fragmentation, overexploitation, pollution, and so on. And all these things cause increased stress in the animals that live in those systems. And if you're more stressed, you're more likely to die and potentially eventually end up in local extinctions of a species as well because of this environmental degradation that you're suffering. So a few examples here. Specifically in the marine environment, we got things like the Phocine morbillivirus, which affected huge numbers of seal populations. When we look at fish in aquaculture systems, we learn more about a lot of diseases that spread in those very heavily kind of compacted populations. Right now, coral biologists are dealing with stony coral tissue loss disease. And then also we've had the big seagrass wasting disease. And in the marine ecosystem, there's certainly been a recent rise in these emergent infectious diseases. And there's a lot of different biological agents that might be responsible for them, for instance, like bacteria and parasites and so on. But viruses in particular, which have actually only just been discovered in like the last century, are really important in the marine environment, but unfortunately, they're also very devastating when they are part of a disease. And of these specific virus diseases, the herpesvirus is the one that we're going to focus on. And there's actually about 100 different kinds of herpesvirus. We're going to focus on one specific one, which is the green turtle herpesvirus, and this causes the disease known as fibropapillomatosis. So you should be able to see here on this turtle, there's quite these large growths that have developed around the flippers here. Sometimes they might be on the eye, here on the neck, around the tails, pretty much in any part of the turtle they could develop. And there's actually quite a few different herpes virus in turtles with lots of different names, but we're going to focus on this one. And just to tell you a little bit about it, it was actually first recorded back in the 1800s, but by the mid-1900s, it had become, it had been observed everywhere from Hawaii, Florida to Malaysia, so really pretty much all around the world. And although we call it the green turtle herpes virus, it actually has been documented in all species of sea turtle, but it's certainly the most common in the green turtle, and particularly in the immature turtle, so the really little guys that are hanging out in these coastal habitats that I described earlier. And although we see these turtles on the exterior of the turtle, if a turtle is actually really in bad shape, if it's to die upon necropsy, they've actually been seen in the internal organs as well. And this clearly is going to be a problem for the turtle because it's going to affect things like their sight. So if you have a tumor on your eye, you might not be able to see your food, or you might not be able to see predators. Also, when these tumors are around your limbs, you know, the turtle is going to have a hard time swimming, for instance. So it affects them in multiple ways. And ultimately, this individual is going to be chronically stressed and immunosuppressed. So that means its stress hormones, for instance, are going to be high. Its immune system is going to be down, and it's not going to be in great shape at all. However, on a positive note, there have been some examples across the world where observed turtles that have been recaptured over time have actually shown to recover from the tumors and recover from the disease as well. Now, the big conundrum with this, this virus and this disease is that the transmission of it is still very unclear. So we still don't really understand enough about how it transmits in between a population, and also what might be the main drivers of the disease as well. And so the objectives of this specific study, following in 2015 this international symposium on fibropapillomatosis, where there was a call really to collect more data on the disease globally so that we could start to figure out some of the answers to this. We wanted to focus on this disease because the Bahamas, like I said, we host a regionally important population of juvenile and adult green sea turtles. And we're very fortunate that there have been in-water studies since the 70s here in the Bahamas focused on sea turtles. And there was an opportunity, therefore, for us to assess what's the spatial and temporal occurrence of fibropapilloma disease here in the Bahamas, and also determine how much of an impact is it having on the green sea turtles here, and also investigate some of the potential drivers of the disease. So the fact that it's not being described here and that we don't know how it's affecting our turtles is really an important question to address. So I'm going to move on to the data collection. So like I mentioned, all my co-authors at the beginning have all got various tagging programs. And this study was possible because we teamed up and pulled all of our data so that we could cover as much of the Bahamas as possible and over as long a time series as possible. So when a turtle is captured, not only do we collect various measurements, weight and so on, if there were tumors, those were acknowledged. More recently now, we've actually started to count the number of tumors and their location on the turtle, the size of them, for so on. Unfortunately, that doesn't exist for the whole data set, just the presence or absence of disease was recorded. We also tag the turtles with these unique numbered flipper tags so that we can, upon recapture, know exactly which individual this is. And we can look at things like growth rates or movements in the turtles as well. And then finally, we release the turtle after that quick workup. Like I mentioned, the revisiting of an area and be able to recapture individuals is a really important aspect of this work. So this is, like I said, happened amongst all our various groups over various time periods and different islands and so on. So what you're going to see now are results from us grouping all of our data together. So that data actually comprised of a huge number of turtle captures, so over 7,900 individual capture events that have taken place. And the sea turtles in the database range from 19 centimeters all the way up to 88. So these are what we call the juvenile and the sub-adult green turtles. Now, just to note that an adult turtle is something over like 90 centimeters or more. So it takes a really long time. Green turtles specifically actually take over anywhere between 30, 40, 50 years to mature. So when you see a sea turtle, more than likely it's going to be like a primary school or secondary school age kind of turtle. We have data from over 18 islands throughout the archipelago, and this data set here that I'm going to focus on spans from 1985 all the way up to last year in 2019. And it is a summation of 1,682 field days, so a huge amount of time out on the water. Now, overall, out of all those captures, only 312 turtles are actually recorded to have the presence of tumors or the FP disease. So this actually only equates to just under 4% of all turtles captured, which to start off with is quite good news compared to some prevalence you might see in other areas. However, this is just overall, so out of all the turtles, and only 312 had the disease. When we start to look at specific islands and even specific locations in islands, this number does go up as the proportion or the incidence of the disease increases in a smaller subpopulation. All right, so first up, I'm going to just show you this figure here. And what I want you to understand from this is that these circles indicate locations where turtles were captured, and the size of them just relates to the number of captures. So the bigger the circle, the more turtles have been caught in that area. And then when we look at the colors, just note that the green circles are areas where no fibropapilloma disease has been seen. And the red colors are increasingly greater proportions of diseased turtles as they get darker. So we can see that areas like Bimini, for instance, and Andros, and this is specifically along the west coast of Andros, have some of our highest proportions of disease present in those populations over there. Following them, areas like Grand Bahama, and the Abacos, Great Exuma, and even down as far as Crooked Island, we've seen diseased turtles over the period. Okay, now to move on and look at some specific numbers here. So this is really just the numbers of turtles caught over time. And this doesn't take into consideration anything like the efforts. Of course, there are more people studying them more recently, and we're spending more time on the water, hence why we're catching more turtles. And so what we did here is break that down to number of captures per day. So we're trying to look at more consistent effort. And we can see we have certainly been putting more effort more recently into capturing these turtles. But if we take that into consideration and look at the mean proportion of fibropapilloma disease that we're seeing over time, we can see that it's actually quite up and down. And the first record of the disease started in 1990. And you can see some of our peak years since then and other areas when there was less. Now, unfortunately, because this study wasn't planned, right, we're making the best of all the data we've accumulated, there is unfortunately not even effort across the islands. So it's really hard to say that, you know, the change over time, because some years certain islands weren't visited and others were, and so on. So that's one of the constraints of this study. When we look at the seasons now, so January to March, April to July, June, July to September, and October to December, we can see that the numbers of turtles that we capture throughout the year vary just because of when we're in the field, but the effort is actually fairly consistent. So when we look at this final graph here on the right, you can see that in the summer months, so July through September, is when we see the peak in diseased turtles, followed by like the fall and the winter, and then fewer in the springtime as well. All right, now if we want to describe specifically these turtles, so here in these graphs, 0 means no disease, and 1 means diseased turtles or presence of tumors. And here on the left, we have the length of the turtle. So this graph is showing that diseased turtles are significantly smaller than diseased turtles. And the width of these boxes just means that we unfortunately have less diseased turtles, or rather, fortunately, we have less diseased turtles than healthy turtles. And in fact, the turtles with the FP are 9.6 centimeters longer. So this means that in general, the really young, newer turtles arriving in the Bahamas do not present with the disease. But following a period of time here in these creeks, they are then starting to develop tumors. And the same follows true for weight. So generally turtles are 6.5 kilos heavier with the disease than without the disease. So the overall point to take from this is just that turtles are not arriving with this disease, they are presenting with it after spending time here in coastal habitats. Now this final graph here just shows you that the body condition of the turtle. So basically, you know, how healthy is a turtle? And there is no significant difference here between the the healthy turtles versus the tumor turtles. And this might mean that our turtles aren't so severely affected by the disease, that they're not actually getting any worse, but also we might expect them to increase somewhat as they get bigger, and they are not, unfortunately. Okay, so I'm going to move on a little bit faster because I have taken up a lot of time. But I want to talk a bit about climate-mediated effects. So aside from human-induced human degradation in these coastal habitats due to our activity, we also need to deal with climate variability, so rapid changes in the climate. And specifically, one of those factors would be sea surface temperature, for instance, especially when we're dealing with marine animals. So this graph is just showing you that over time, our sea surface temperatures are becoming higher, greater, and there's also been an increase in heat waves, in frequency and length of heat waves in the ocean as well. So our oceans are certainly getting warmer. And so we wanted to look at this because we know that a lot of marine organisms have been shown to become less healthy. So they become immunodepressed, for instance, and more susceptible to disease in warmer waters. For instance, corals are a great example of an organism that is suffering due to higher water temperatures. And also, we know that pathogens will replicate faster in warmer habitats as well. So there's this two angles or two aspects to this. So when we look at there, like the Bahamas, with these amazing shallow banks and deeper water habitats, there's a lot of variation in the sea surface temperatures that we would encounter. And because sea turtles are ectotherms, so they're influenced by the external water temperature, this is something that we are certainly interested in. So for instance, I'm just going to point out, we've got the tongue of the ocean and the Exuma Sound, these deeper bodies of water, and therefore also obviously the Atlantic, are more stable. So the deeper the body of water, the less change we're going to see in the water temperature over the course of the year. Whereas the bank systems, because they're a lot shallower, there's a lot more extreme variation in the temperature of the water based on tidal movement or even day-night changes as well. So now if we look back at a figure of the country, you can see here, we'll just go back one time, you see where the deep bodies of water are. You can see here, based on this figure, the color variation is due to temperature variation. So these deeper bodies of water are the cooler blues because there isn't a lot of temperature variation, and the shallow banks have a lot more variation. And this figure is showing you two things. So where is there more variation in the marine environment, but also the circles relate to human population here in the Bahamas. So the bigger and darker the circle, the more humans we have living in an area. And the reason we looked at this is because our human population is potentially having an impact on our sea turtle disease. And we decided, no, that is most probably not the case, because it's certainly when we look at areas like, for instance, the west coast of Andros, where there is a great proportion of diseased turtles. If you're fortunate enough to have been there, you'll know there's very few people around, not for miles. Similarly, in an area like Bimini, there are not many people, but we're seeing big proportions of the disease. And so moving forwards, we're just going to focus on the actual sea temperature variability. So this is just a reminder again of where the disease is present and not present, and to highlight again these areas that are in green where there's not very much, no disease at all, are more islands that are much more kind of encompassed by deep bodies of water. And so when we looked at the captured temperature, so we used satellite-derived temperature data, we could see that healthy turtles, remember the 0 is the healthy turtles, they tended to be captured in waters that were almost 2 degrees cooler than the temperature of the water where diseased turtles were being captured as well. And that was a significant difference there. So that's what's led us now. And I can't include all the results here today, but this is where we're looking at now. This again is an overall of all the Bahamas. Each circle here is a capture location, and the color of that circle, the darker it is, the more variability there is. So if we look at some of these darker circles here, for instance, Grand Bahama, the west coast of Andros, there could be anything from 10 to 12 degrees centigrade change in water temperature there over the course of the year. And that's a huge change to go through. Whereas if we look at some of these more kind of oceanic islands in the south, like in Iguanas, there's very little variability, between 3 to 6 or 4 to 6 degrees centigrade. And so we're looking further into this. Now, this is overall. When we start to go into a specific island, we can look in much more detail. And we know from just preliminary findings that in some of these habitats, warmer temperatures are getting warmer and colder temperatures are getting colder. Therefore, the variability is increasing. So that was the final result I wanted to mention. There is increasing and decreasing sea surface temperatures depending on the island, and therefore greater variability. And this dynamic water environment that the turtles are spending time in can lead to greater physiological stress, unfortunately. And that we believe this may be what is potentially driving this variation in the presence of disease across the islands. So I wanted to finish there talking about that. As I said, these are preliminary findings. We're excited to be wrapping up this study very soon. I wanted to just highlight very quickly some of the other threats that unfortunately our turtles are facing, such as entanglement and fishing line, can result in injury, even death. We're still dealing with things like habitat destruction. So this is, for example, Lighthouse Point in Eleuthera, which is soon going to be looking like this. And when we do this to these kind of environments, the water quality is seriously affected, and therefore marine organisms are negatively impacted. When we go and put structures where they shouldn't be, increasing storms, so our increasing severity of hurricanes, for instance, are damaging habitats that are being used for nesting. So this is an example of a dune that was washed away due to severe storms, and therefore a turtle nest was exposed and lost. We also still unfortunately see huge amounts, not huge, I'm sorry, but significant amounts of poaching in certain areas of these endangered species. And so finally, I just want to highlight the Bahamas Sea Turtle Network. You can find us on Facebook, but we really rely on not only the partners, but also the citizens here in the Bahamas or visitors who are traveling through to report to us anything they're seeing. And it might be you're seeing nesting of turtles. We'd love to hear about it. So taking a photo of the nest, telling us where it is, the tracks, we can determine things like species. Like here, this is a loggerhead sea turtle that are nesting right now. Between kind of March through August, we'll be seeing increasing numbers of this. So please let us know. Let us know if you're seeing disease, poaching, and so on. Just report anything you can to us. It would be much appreciated. And just to finish up, to say thank you very much to, again, all the partners, all the Bahamas Sea Turtle Network partners. And thank you for having me. Apologies, I went quite over time.
Okay, that was an absolutely fantastic presentation. Thank you so much, Annabelle. Just to let everybody know, you can ask questions. There's a question and answer box, and we will have a question and answer session after everybody has presented, after all the panelists has presented. So feel free to ask as many questions as you would like. Please put them in the question and answer chat box, and we will get to them once the presentation is over. Thank you. Coming up next is Mr. Joseph Wazalewski, who is the president of the wildlife consulting firm Natural Selections of South Florida. And he will be presenting on the rapid influx of invasive reptiles throughout Florida, the Caribbean, and the Bahamas.
Am I on, Scott?
Yes, you are.
Oh, so I'm ready to go?
Yes, sir.
Okay. All right. It took me just a couple seconds to figure out. But I have it on, and you see my PowerPoint up on the screen?
Yes, we do.
Okay. Then let's begin and talk about this issue that's happening all around, actually, the world, not only Florida, Bahamas, and the Caribbean, of invasive species. And I'm going to talk mainly about reptile species. And wait, let's see, where am I at? And I just want you to look at my little map of the Bahamas here. And I want to talk about one island, a small island just south of Bimini. It's about 15, 16 kilometers south of Bimini. And This particular island is about 90 hectare, and prior to 2004, there were no green iguanas. At around that time, 4, 5, or 6, someone released it would have had to have been a pair or a gravid female. And over time, people did not mind seeing an occasional iguana, but the numbers began to grow to a point where I was asked in 2014 to take a trip to the island and do a quick survey. And it was like a green wave as I was driving along a golf course on a golf cart, a green wave of iguanas. There was literally thousands of them. So this has been updated. I've been there more, but just to give you a little idea about numbers, 22 trips, 6,374 iguanas were removed. 3 people a trip, 3 days a trip. We average about 5 days of catching iguanas. It's pretty tough work. You could do more, but you kind of wear yourself out. Anyway, if you take all of that and 900 man hours, we figure we're catching 16.5 iguanas per hour. That's an amazing number. And what are the iguanas doing wrong, everybody says? Well, let's begin with FAA, Federal Aviation Administration. And iguanas on a big iguana and a small plane can be a disaster. They're actually tunneling under runways. And in Puerto Rico, they've actually collapsed highways. So FAA safety hazards. People are into their landscaping, and this is beautiful landscaping on Cat Cay before green iguanas. And this is what it looks like after green iguanas. They are vegetarian and they do eat all different species of plants. However, being a boreal, we're finding out more and more that they will and do, in fact, eat many different birds' eggs and young birds that are nesting up in the trees. And in Florida, in the Florida Keys, we're beginning to find the native tree snail in stomachs of iguanas. So, you know, not only do they eat the vegetation, but they could be detrimental in other ways. How do we catch them? Out of, well, right now we're over 17,000 iguanas, 5,000 iguanas in Florida, and miscellaneous other hundreds of iguanas. We catch, I would say 95% by hand or by tongs. And when they're way up in the trees, I kind of call this flying iguanas because we just take a painter's pole and sort of annoy them enough that when an iguana seeks its escape, it just dives right out of a tree and it hits ground running. So our best bet is to catch them in midair, or lately we've been using a landing net. And for the occasional iguana that we don't catch, we can run them down, usually. We also noose them. This might look bad, but the iguana is not hurt at all. You slip the noose over its neck, it walks into it, and you're able to remove it that way. Traps. A lot of people talk about traps. To me, they're time consuming, and we're catching more iguanas by hand. However, if you own a hotel or a home and there are a couple of iguanas that are becoming a nuisance around your pool or whatever, setting traps could very well be useful. So don't ever discount that. Here's the big question: euthanasia. It's sad to say, because nobody likes iguanas more than I, maybe Scott and Shannon, but that's okay. But these animals need to be euthanized. There's no other way. And if somebody can come up with a better way, just let us know. And just watch it, because the next couple slides could be some graphic images. We use captive bolt systems. What do you call it? It's called blunt head trauma is what's accepted. Just to let you know, here's several iguanas from a night's work. And again, we're pretty effective at this, my team. Every single iguana out of those 20-some thousand are weighed, measured, and sexed. And believe it or not, right out of all the entire populations that we've removed, it's right at 48%. It's right there, right close to being in the middle of males and females. We open the females during all times of the year to check their reproductive status. And in the fall, October, November, their fat bodies develop. And around January, February, follicles begin. And this is April is when they nest. So in addition to, I should have said it earlier, to removing 20-some thousand iguanas, we have removed probably around 10,000 eggs. So let's talk real quick about Florida. 56 plus or minus, because we published a paper on that a few years ago, and the numbers were increasing as we were writing. 3 frogs, 4 turtles, 5 snakes, and 44 lizards. I always mention about the Bahamas when it comes to Florida, and you'll see in a moment or two. A few of these invasive lizards that are found in Florida are also found in the Bahamas, like this spiny-tailed iguana is found, they're from Central Mexico and Central America. They're found in the Berry Islands right now. And I've been hearing a couple of reports of different areas in the Bahamas that we need to look at. So that's spiny-tailed iguanas. This is an animal that's found, we're catching by the thousands in South Florida. They're native to Argentina. They're omnivorous. They'll eat most anything from eggs to cocoa plum berries to vegetation to carrion. They'll eat a little bit of everything. The state of Florida has removed thousands of these. These animals would really be bad news if they got to the Bahamas. By the way, this is just a shot of an alligator nest that we put 3 trap cameras around, and there were 33 alligator eggs in the nest. And over time, we estimated, we didn't estimate, we saw 4 individual tegus go and eat every single egg. So that's a danger all over. In Florida, we have chameleons from Madagascar that get huge. We don't really know the impact of these, although they're insectivorous, and there has to be some long-term impact that we will figure out someday. This is the next, if any invasive species comes to the Bahamas, this is going to be the next one, called a red-headed gomet. And the reason I say that is because they're very common in South Florida. They've only been around for about 10 years, and they nest in nice soft soil, and they're known to nest in pots that are plants, nursery pots. So a planter, can ship these pots over anywhere, and these eggs can hatch, and that's how they get around. Here in Florida, we have removed 3 Nile crocodiles so far. That's scary enough, but the huge possible impact is crocodiles are known to hybridize. The American crocs and the Cuban crocs hybridize in Cuba, and in Mexico, the morlets hybridize with American crocs. So it would certainly, it could happen if Nile crocs got to Florida. I don't know how many of you folks out there have heard of the Burmese pythons in South Florida, the population. Never did I think I'll be showing a range map of Burmese pythons in Florida, but they're here to stay. And thousands have been captured and removed from the Everglades. We actually, not in 2016, we brought in 2 Irula Indians from India, along with Ram Whitaker, snake experts, to show us how to locate pythons so we can catch them. And the biggest one we caught was like 17 feet long. Just to let you know, they are a danger, and this is, believe it or not, a full-grown deer being taken out of a stomach of a python. And this was in the early days, and since that time, we know of at least 8 to 10 occasions where there were deer or parts of deer in stomachs of pythons. New age of python hunters. There are 100 contracted people, including a lot of ladies out there, that are catching pythons in the Everglades and getting paid to do it. I think it's so far between 5,000 and 6,000 of these pythons have been removed from the Everglades. And you can see some of these hunters. What we've been finding lately is they're going out to sea. The top right corner photo was sent to me a couple weeks ago by a couple that were out on their boat several miles offshore in the Florida Keys. And the one on the bottom is on a weather station in the, I'm gonna have to say 2 miles out. And the one around the pole, I guess, was just tired of swimming and it just stuck on a pole. So they could potentially raft over to the Bahamas. And getting to the Bahamas here and all of these invasive species, let's talk about the proximity to Florida. Daily flights, boats, containers bringing goods. And I already mentioned palm trees, nurseries. Marine toads are hiding in pots. Agama lizards are nesting in pots. So it is ultimate of importance is biosecurity, should be included in any future environmental budget. At this time, there's 26 to 30 species of invasive Bahamian herpetofauna. 3 turtles, 7 to 10 lizards. We're still kind of out on the fence on that because there's a few records of them, but not authenticated. Snakes and frogs. So why is all of this important? You're going to see a little bit later with one of my colleagues giving a talk that in 2016, another one of my colleagues, Mike Valley, captured hybrid iguanas between Cyclura species and green iguanas. So that is kind of our one of our biggest fears, especially in the Bahamian archipelago, with 3 different species of native and endemic iguanas, totaling 7 taxa. If these green iguanas got to islands where the native iguanas live, it could spell disaster. So what is the solution? Legislation, permitting, limiting ownership. In Florida, as of yesterday, today, July 1st, it is illegal to own green iguanas and tegu lizards. We should develop programs for removal. We need to keep pressure on these invasions. And once again, I'm going to say this, and I'm going to say it over and over again, is any future budgets should make biosecurity a plan and be consistent with that plan and use a lot of funds for this, because that will help stop this invasion. After biosecurity, rapid response. If someone sees a green iguana, to notify the authorities, and we need to get out there as soon as possible to take care of the situation. There's a whole bunch of people and universities and agencies that I've been working with all along with this issue. And I just want to thank everybody and thank everybody for listening. The end. And I guess we'll have questions later.
Yes, we will have questions later. Thank you so much, Joe. That was a very informative talk, and it's also very alarming, especially seeing how there's so many non-native herps, which are reptiles and amphibians in Florida. And because of our close proximity, as you mentioned, we are always under the gun for an invasion of a new species to come in. And so that is alarming information. And I hope that everyone listening will also be keeping an eye out for any new species or anything that they may see, because that's very important. That's important information for us to know as well. And so before we move into the next presentation, I just want to highlight.
Okay.
Okay. Okay. I just want to highlight the Natural History Field Guide of the Bahamas. Some of you may have heard about this book, but just in case you haven't, the Natural History of the Bahamas Field Guide is small enough to carry on trips, and it is an ultra comprehensive guide of terrestrial and coastal flora and fauna, and includes species and common names, locations, and detailed descriptions of these animals. The book was written by experts in Bahamian ecology, with contributions by more than 30 other naturalists, myself included, and covers organisms studied in the Bahamas and the Turks and Caicos Islands, with more than 700 color photos. It is available for sale in New Providence at Logos Bookstore and Media Enterprises, as well as all national park offices that the BNT manages throughout the Bahamas. The book can also be purchased online at Cornell University Press and online book retailers such as Amazon for Kindle, instantly and by mail. So and most importantly, all royalties from book sales benefit the Bahamas National Trust. So it's a great way to support the BNHC and the work that we do. So I encourage you all, if you haven't gotten this book, a copy of this book, it is absolutely fantastic. And it is up to date with all of the latest information that you would want to know about the Bahamian flora and fauna, or as a lot of the Bahamian flora and fauna. So I really encourage you to look into purchasing this book. Okay. And with that, we will move into our next presentation. And it is by Miss Shannon Yates, who is the Assistant Curator of the Retreat Gardens National Park on New Providence. And her presentation is entitled The Green Iguana Invasion in the Bahamas. So Shannon.
Okay, can you guys hear me now? Hello?
Yes, we can hear you.
Okay, good. So invasive species introduction constitute a significant problem around the globe. It threatens biodiversity and populations of native and endemic organisms. The one we are focusing on today is the green iguana. And just for context, Joe already mentioned this, but we have about 30 different species of reptiles and amphibians that have been introduced to the Bahamas directly as a result of our proximity from South Florida in shipments of construction and even landscape materials such as plants. Anthropogenic species, anthropogenic invasive species like the green iguana are expanding their distribution to the islands that are home to endemic rock iguana species in the Bahamas. So the common green iguana looks like this. So it has a long tail. The tail has black bands on it. The cheeks of the iguana is flat. It has spine under its throat, and they're great swimmers. They're also arboreal, meaning that they live in trees, and their general diet consists of leaves and vegetation. So where are these guys from? Their native range is southern Mexico to central Brazil, Paraguay, and Bolivia. They are native to some Caribbean islands, specifically Grenada, Aruba, Curaçao, Trinidad and Tobago, St. Lucia, St. Vincent, and Utila. They've been introduced to other Caribbean islands, including the Bahamas. So what does our native rock iguana look like? So this is the Andrus rock iguana. They are large terrestrial animals. The largest of our Bahamian iguanas is from Andrus in the photo. They prefer not to swim, but if needs, they can swim. They have no bands on their tails comparing to the green iguanas. The body is generally brown. Their throat has no spike and is smooth. Our Bahamian rock iguanas vary in coloration, as you can see in this other slide. We have 7 species and subspecies throughout our islands. The Allen Keys in Exuma, San Salvador, White Key, Acklins, and Crooked Island. And you can see the variation in different colors, including the San Salvador and the Acklins iguanas. So why is this a concern for us? Well, on New Providence alone, we have had 23 different sightings of green iguanas from 1996. And these have been documented by Ms. Sandra Buckner, who have been documenting the herp fauna of the Bahamas from the late 80s into the early 1990s. So as I mentioned, we have 23 different sightings from 1996 on New Providence, including Paradise Island. These are some of the individuals that were caught on New Providence and brought to the Bahamas National Trust, which was euthanized. We have had females that had eggs in her, meaning that we suspect that they are reproducing on New Providence, and large males of reproductive age. Throughout the Bahamas, we've had confirmed sightings on Bimini, which Cat Cay specifically, which Joe himself has been working over the years to control the population. We've had recent sightings in Grand Bahama, Abaco, Eleuthera. There has been one on Daniel Key Exuma and a key off San Salvador, and most recently Andrus. Now these are of concern to us because on the islands of Andrus and Exuma, we do have native populations of rock iguanas. And in 2016, something happened that we never thought that could ever happen. Conservation biologists confirmed the hybridization of rock iguanas with green iguanas. So in the Great Cayman Islands, Cyclura nubella caymanensis, which is the sister island iguana, was confirmed to hybridize with green iguana. And Cayman Island has a large population of green iguanas which they have been culling and trying to keep under management since the early 2000s. So 6 hybrid iguanas were documented from that population, and this is the comparison of what the native rock iguana looked like versus what the green iguana hybrid looked like. And then a comparison between the green iguana hybrid and the green iguana juvenile. All right. So that ends my presentation. I'd like to thank Ms. Buckner for giving me the information on where all the confirmed sightings were.
All right. Thank you so much, Shannon. Another wonderful presentation, and also very thought-provoking, seeing how these animals are spreading over New Providence, and it's almost like every year we're seeing more of them pop up. I think earlier this year, one of my friends found at least 3 iguanas in his area, and then I recently got some more calls about green iguanas out in Arawak Key. It is alarming to see these animals all over the place right now. And the last thing we want is to have them hitting these islands where these iguanas are, where rock iguanas are, and hybridizing with them, because then that's that's a really, really serious problem. So before we move into the final speaker for the afternoon, I just want to say again that the session is being recorded. So our BNHC V sessions are being recorded and is streaming live on Facebook. And after today's sessions, the recordings will be available to all by visiting the Bahamas National Trust's Facebook page and YouTube channel. If you would like to keep up or get in touch with the BNHC, you can do so by visiting our website, which is bnhc242.com, and facebook.com/the BNHC to follow social media updates. And you can also send questions and suggestions to our email, which is bnhc@bnt.bs. Okay. And so the final speaker for this morning is Dr. Nathan Robinson, who is another adjunct scientist at KB Luthra and the Oceanographic Foundation in Spain. And his presentation is entitled Assessing the Short-Term Impacts of Biologgers on Juvenile Green Sea Turtles using a combination of animal-borne cameras and drones. So without further ado, Dr. Nathan Robinson.
Hello.
Hello? Yes.
Oh, perfect. Just making sure I have everything working. Thank you so much, Scott, and thank you for the opportunity to present today. It's wonderful to see everyone adapting to the current crisis and taking things online. I think it's a great way to get as many people exposed to some of the great research and work that's going on in the Bahamas. So kudos to you guys. I also want to give a quick little shout out to the other talkers and the presentations. It's been absolutely fantastic. Very fascinating stuff. So it's an honor to finish this turtle iguana sandwich with the final turtle talk, where I'm going to be talking about the impacts of, yes, biologgers on juvenile sea turtles. Now, I'm sure that over several of the other talks that have been given in your different sessions, lots of people have learned about the fantastic insights we can gain through the use of biologgers on various marine animals. But I'm hoping to kind of flip that idea a little bit on its head and start to look at how these devices that what we're using to gain insights into these animals, how they're actually impacting the animals themselves. Right, so for anyone who's already lost by what I'm talking about, a biologger is a sensor, a data logger that is mounted onto your animal of interest. There's a big range of different potential biologgers out there. There could be satellite transmitters that tell you where animals move. We're going to be talking today about animal-borne cameras. So these are devices that give you a first person's perspective into how an animal sees its environment, but it can be anything from temperature sensors to dive sensors to you name it, someone's invented it and strapped it on an animal. What's so fantastic about these devices, especially for the marine realm, is they allow us to get new insights into animals that would be very difficult to track visually. So you can think of lots of terrestrial animals, and if we want to study them, then we can generally kind of follow them quite easily. But with the marine animals, surface, we don't really know where they are. It's almost impossible to track them. But by actually deploying the data logger onto the animal itself, you get this wonderful insight into how these animals are living their lives. These devices, because of the insights they can provide, and because they're continually getting smaller and cheaper, thanks to technological innovation, their utilization, their use is increasing dramatically. I think the latest estimates is somewhere between 10 to 20% increase in the number of studies using biologgers in the realm of marine biology every year or so. So very rapidly increasing. Now, as we're using more and more of these devices to get insights into these animals, there becomes a bigger and bigger need for us to start to look back at some of our work and think, what are the impacts of these biologgers? And specifically, I'm going to be talking about the impacts of biologgers on sea turtles. I won't give an introduction to sea turtles because Annabelle's already covered that wonderfully earlier. So I'll just jump right into the logistics. Now, when we're talking about the impacts of, say, a biologger, we can generally divide it into short-term impacts and long-term impacts. Well, the long-term impacts might be something like discomfort. You might have a device that's attached to an animal that could in some way, if it's not put on properly, impinge the movements or just be a bit of an irritation to the animal that has to lug this thing around. Similarly, they can also affect hydrodynamics. Most marine animals have evolved to glide through the water column, and if we add something onto their carapace, in the case of a sea turtle, or onto their fin, in the case of a dolphin or a shark, then we can start to affect on their hydrodynamics and potentially increase the drag, increase the cost it takes them to swim forward. There's also short-term issues, and the short-term issues generally revolve around capture and handling stress of the animals we're studying. If we want to deploy a biologger, you generally need to have an animal in your hands so you can attach that device, and that can be relatively stressful. Generally, it will elicit some kind of predatory or flight or fight response from that animal. If we start studying these factors, we start studying the potential impacts that handling or retention of handling an animal or retention of a biologger might have on them, they can provide some great insights. Importantly, they can provide us some way to kind of judge the ethics of tagging or animal handling. I'm sure that many people who are listening to this talk agree that the data that we've been collecting on sea turtles or iguanas or many other species that have been discussed in these talks are of value. It can actually help conserve these species. But if we're also having a potentially negative impact, increasing the stress on these animals, then where's the balance? And by getting this information, we can start to figure out where that balance should be. What that balance should be. Another important factor is to do with how do we interpret the data that they're produced from these biologgers. If we have an animal that is, say, stressed because it's just been handled, it's not going to be exhibiting the same behavioral patterns as an animal that may not be stressed. Now, if we're using these biologgers to get insights into the behavior of sea turtles, for example, we might not be able to use those data because it's not going to reflect an animal in the wild. It'll reflect an animal under stress. So for those reasons, very important to start to understand the potential impacts that biologgers can have on wild animals. Now, if we wanted to understand that impact, an easy way to do it would be compare the response and the behaviors of animals, say, with biologgers to animals without biologgers. The problem is, as I mentioned on my first slide, how do we start recording data on wild animals that are difficult to track? I mean, it's very easy to get data on an animal with a biologger. That's the data that's provided by the biologger. But how do we provide that comparison to make sure that we know that this is data, the biologgers are providing data that is how to compare whether you have, say, a disturbed animal or a non-disturbed animal. And for that, I want to quickly introduce another potential research method, and that's the use of unoccupied aerial vehicles, or drones. Now, I don't really need to give much of an introduction into these devices. They've been kind of rapidly commercialized in the past decade, and now ecologists, researchers, hobbyists all around the world have access to drones. I'm sure many people who are listening to this talk have either used one or have friends who've used one for whatever reason. Well, ecologists, we've jumped on this technology and we've rapidly incorporated it into a lot of our projects. And they're very flexible tools. The drone that we've been using in this study is called a quadcopter. They tend to come in two different designs. Some designs are called fixed wings, and they look like small airplanes. And then there's the quadcopters that look like miniature helicopters. The nice thing about the quadcopters is they allow for flexible flight plans. So you can fly up and kind of move wherever you want to go. This means you can track an animal quite happily using a drone from an aerial perspective. In addition, several studies have shown that when drones are flown sufficiently high enough, they have minimal, if any, to the animals they're trying to study. So we can actually start to use footage from drones to get information on the natural behavior of these animals, and then we can compare it to animals that, say, have biologgers, and compare the insights between both methods for collecting data. So with that, I want to reiterate my objective for this study. And the objective of the study was to assess the effects of deploying biologgers on green sea turtles. And I was going to do this using a combination of animal-borne cameras. So these are cameras deployed onto the sea turtle. It's a first-person perspective of how they see their life and how they see the world around them, and drones, which give, say, a third-person perspective that allows us to monitor these animals over time. The question we wanted to ask is if it's having an effect. And because of that, we can come up with a couple of simple predictions or hypotheses, which, if proven right or wrong, might be able to answer our question of whether or not we're having an effect. So number one, our first null hypothesis was turtles' behavior does not change after biologger deployment. This idea is if we're going to find a turtle and then deploy, say, an animal-borne camera, if you're thinking you're talking about a stressed animal, you'd expect that the behavior of the animal would be different over time. Over time, the animal might become more relaxed, less affected by the initial capture, stress, and so its behavior patterns would change. If the animal isn't affected at all by the capture, it would immediately just go back to doing whatever it was before you caught it, and you wouldn't see any change in its behaviors over the time that camera was recording. Our second hypothesis that we wanted to test was that turtle behavior with or without biologgers be comparable, would be somewhat similar. So what I'm trying to say through that hypothesis is that turtles who are animal-borne cameras, so turtles with biologgers, would be conducting very similar behaviors to turtles that don't have biologgers, that haven't been disturbed, but are being recorded by drone. To answer this question, we conducted this project in Eleuthera, to the east of the Bahamian archipelago, and we were working primarily in several beautiful shallow mangrove creeks on the southern end of the island, particularly Starved Creek, Rollins Creek, and Deep Creek. When we would head out into these systems, we would head in a small boat looking for evidence of a sea turtle. When we found a sea turtle, We'd follow it by boat for 2 or 3 minutes until the turtle got tired. We'd then send a snorker into the water who would grab the turtle and lift up, we'd pick it up by the boat. We would then attach our animal-borne camera, which you can see in the bottom left-hand image. Then we would release the animal back into the water. Now, heading back to that image of the animal-borne camera, there's a couple of things I want to point out, and primarily these little metal links where it says galvanic timed release and has a little arrow. These are corrodable links, which means that after 3 to 4 hours, which is the amount of time that the camera is able to record for, they corrode, they pop off. Then the camera, which has a buoy, a little float attached to the top on a radio transmitter, will float to the surface. We'll be able to pick up the image from the signal, from the radio transmitter and recover the camera. So this is how we can actually recover the camera, recover the footage without having to recapture that turtle. Then for the drone part of the study, we would conduct drone surveys in the same creeks, but on different days to when we were deploying the cameras. The idea being we didn't want to start recording animals by drone that might already been disturbed, the boat or our actions in the same habitats. We'd head out there, we would fly the drone at a height around 30 meters until we spotted a turtle. When we spotted a turtle, we'd lower the drone to 15 meters. We picked that height because other studies out there have shown that sea turtles of comparable sizes don't respond to a drone as long as it stays above that 15-meter height range. And then we recorded continually until the drone ran out of battery, which was generally around 15 minutes. After that, we now have 2 sections of footage. We have 3 to 4 hour chunks of footage from our animal-borne cameras, and we have the footage recorded by the drone. And what we did with the animal-borne camera footage is we further divided it into 30-minute sections. So that 4-hour chunk was divided into 30-minute sections so we could look at behavioral changes over time. Then we reviewed all this footage and we started to quantify the percentage of time spent conducting different behaviors from both types of footage. Now, the different behaviors that we're categorizing, I'll show you in a second. And this is a clip from our turtle cam. This is the moment everyone's been waiting for. So this is an example of turtle swimming, and that's one of our categories. We also have breathing. We also have feeding, turtle feeding. We have resting, so the speed when the turtle's just sitting on the seafloor or sitting at the surface or wedged under a rock where they often relax. And here's another fantastic, very engaging behavior we often respond, which is social behavior. Turtles interacting with one another. Over the past 2 years, we've now deployed and recovered over 70 animal-borne cameras. We've conducted over 40 drone surveys, but as you can imagine, that's a lot of footage to process, and we're not done with it yet. So what I'm going to present today is just some preliminary results from 10 animal-borne cameras and 10 drone surveys. And we only, even though we actually have data from 3 regions, we're only going to focus on a single region, and that's Starved Creek. And these are the data that we found. Now, to explain this rather complex graph, first I want to point you to the axes on the left, the y-axis. It's time spent during different behaviors. So this is the amount of time the animals spent, or those animals collectively conducted each one of the behaviors that we identified. And the different colors of those bar charts reflect a different behavior. And you can see those behaviors on the right-hand side. Blue being swimming, black being time spent at the surface, yellow being resting, green being foraging, red being interacting, and white being other. So anything that didn't fall under those categories. At the bottom of the graph, you can see time since deployment. So that first bar would be the first 30 minutes that we were recording from the animal-borne camera. The next would be the second 30 minutes, and next will be the third 30 minutes, and that goes the whole way along until you see that dotted line. And then that final column that's labeled drone, that's the behavior that we identified from the drone footage. Now, there's a lot I could say about what's going on here, but to keep it simple, I want to go back to our initial hypotheses and see if we've seen our predictions. So our initial null hypothesis was turtle behavior does not change after biologger deployment. And if we look back at the graph, which I've now compressed a bit and put to the right-hand side, you can see that that's not true. If that was true, all those bars would look very much the same, but we actually see some distinct patterns. And what's interesting is in that first 30 minutes, the turtle swims almost nonstop. And then over the next hour to 2 hours, we see an increase in resting behavior and a decrease in swimming. Then after about 2.5 hours, we start seeing an increase in swimming behavior again, and resting tends to decline. So this is probably, once you let go of the animal's first response is a flight response. So it's just trying to get away. It then has to take up to an hour and a half to 2 hours to start to recover itself. And then after that time, that's when it starts going back to its normal behaviors. But it is very interesting to see that even in that first 30 minutes, we start to see some essential behaviors, mainly foraging. So it's interesting to see that even though this animal might be under a little bit of handling stress, they still seem to be able to parcel time aside for eating. It's not distracting them from feeding. Our second hypothesis was that turtle behaviors with and without biologgers would be comparable. So to answer that, we've got to look at the right-hand side of this graph, and we can compare the drone, that being our control for turtles without biologgers, to everything else. And what we find is very interesting is that actually at the 2.5 to 3 hour mark, the behaviors seem very similar, while elsewhere, there's quite a big difference between the behaviors of the turtles recorded by drone and the turtles recorded by the animal-borne camera. So this is suggesting to us, at least from these preliminary analyses, that after about 2.5 hours, these animals are starting to resume their natural behaviors once more. So what does this mean? Well, it's clear that we do have a short-term impact on the behavior of these animals through capture and handling stress. Yet, at the same time, it does seem that the essential behaviors, feeding and socializing, seem to continue even from immediately after these animals have been released. So, because of that, it might be that the negative consequences of handling stress of these animals and biologger deployment may be relatively minimal. That said, I do want to say that these are preliminary conclusions. We're still in the process of analyzing all our data, and we are talking about animal behavior, and there's a huge amount of variation in the behavior of different animals. So I'm sure as we go through the rest of those 70 turtle cams and there's 30 remaining drone surveys, new patterns will come to emerge, but that's what we see. Another thing we also want to do with the data is there are other ways that we can start to analyze these data to look at other behaviors. We can start to look at, say, diving behavior. Maybe we'll see subtle changes in how frequently a turtle dives. And we can once again compare the drone footage to the animal-borne camera footage and see if we can parcel out some of these other behaviors. Or maybe dive behavior doesn't change at all. So dive behavior stays the same while their behaviors in terms of whether they're feeding or swimming or resting actually start to change. So there's still other ways that we can analyze these data. So I just want to say, stay tuned because there's plenty more to come. And I'll end, because I'm pretty sure that's what most people are here for, with even more turtle cam footage. So this is a quick best of round of some of my favorite clips that we've got from our animal-borne cameras. First off, just the beautiful crystal clear waters in the Luthra always kind of take my breath away. Here we have some great footage of another green turtle munching on some seagrass. This is a whiptail stingray cruising past, and that's actually a hawksbill turtle. That's a slightly different species. Another thing I think we're looking at using these cameras for is actually using these animals as surveyors. So you can actually start to identify the different habitats through the turtles. And this is my favorite clip of our whole footage, just showing that sea turtles don't really respect social distancing and personal space the way that most of us are doing a great job of these days. Okay, with that, I have to thank all the incredible, wonderful people. There's some of them who are listening to this talk right now who have been part of this project. It's been a huge effort from far too many people to mention here, but they've all been absolutely essential to making this project a success. I also have to thank all the different organizations and affiliations for the authors of the study. We got fantastic funding from Paralens, British Colonial Group, and Finding Our Way, supported by NOAA to help make this project a reality. Thank you again for Bahamas Natural History Conference, providing an opportunity to talk. And I'd like to end by giving a quick thank you to 60 Second Docs, Ocean X, and Great Big Story, who have all done short videos online recently on the Animal-Born Camera project. Has been amazing to conduct kind of public outreach on such a big scale using this project as a platform. With that, I think after I finish talking, we'll be moving on to questions. But if anyone has any questions they want to ask me directly or don't have enough time now, there's my email address and my social media if you want to see more animal-born camera footage at any time. So thank you so much for your time. I hope I didn't go too much over, and I'm excited for people's questions.
All right. Thank you, Dr. Robinson, for that amazing presentation. In fact, thank you to all of the panelists for those fantabulously awesome presentations. I thoroughly enjoyed it, and I'm sure that everybody else has. I just want to say that we hope that you can join us for the remaining sessions scheduled to continue every Wednesday into August as a part of the BNHCV. As always, we welcome your questions, feedback, and support. And as the BNT team continues to pivot our programming towards this new normal methodologies, including changing the platform of our conference to a virtual reality, there is still a need for funding events like these weekly sessions. And we would be grateful if you consider donating towards the success of BNHC 2020. If you are enjoying the BNHC virtual webinar series, we simply ask that you consider giving a donation in any amount as a means of showing your support. An email with a donation link will be sent following this session. So I want to say thank you all. And yes, it is the question and answer period. And we have a ton of questions. So, oh boy. Okay, panelists, prepare yourselves. I got a bunch. And so let me see what I'm going to start off with a question directed to Annabelle. This one was given to me. It says, do you think the number of turtles seen with diseases coincides with breeding season? And is that one way in which the disease is being spread?
Thanks, Scott. So there's quite a few things I wasn't able to include in my study, but these turtles that we're seeing with the tumors are all juvenile, first of all. So they haven't reached sexual maturity. So then they don't really have any correlation with nesting season. And also they are here year round. So they will be really tightly kind of attached to a very specific creek. So for example, a lot of the locations where we study these turtles, they are only ever recaptured in the exact same creek. They won't even move to another creek, not even like a kilometer down the coastline. So they're really highly slightly attached. So there's not a lot of movement between these little subpopulations. And they're there year round, like I said, potentially up to even a decade until they get bigger and can move into deeper water.
Okay. All right, so I have another question from Anselino Davis, and it is, are any of the turtle viruses contagious to humans?
That's a great question. No, it's not something that we need to be concerned about as far as when we're handling these organisms or anything like that, or swimming in the same kind of water with them. It's not something that we're not a host for that particular strain of the virus. So even though it sounds, like herpesviruses, there's many of them, like I said, in the talk. So there's lots of different strains that are going to affect different organisms, including humans, but they're not transferable.
Okay. All right. So another question is the lower seasonal percentage of fibropapilloma why can't they say
FP, to say FP, Scott.
Yes. Okay. Of FP occurrence related to demographic ratio differences, or does the disease have seasonal flare-ups?
Yeah, so that was what I was trying to get at, is that because these turtles, first of all, they are there year-round, what we're seeing is more like a seasonal flare-up, and most likely due to the water temperatures. So the fact that we see that spike in summer and also in the wintertime when you get the other extreme of water temperature for high and low. But also, these turtles are obviously, because they're there year round, these tumors, what we still don't really understand is, how long they take to, first of all, present themselves, but then also grow. And some of the things I couldn't include in the talk is the fact that we found turtles which one year were healthy. We've returned the next year, might have had over 30 tumors. I've also tagged turtles with tumors and not found them again. And we have not seen any turtles that have regressed though. But yeah, so there's lots of different factors there, but certainly the flare-ups in the summertime are most likely due to this increased stress, because you get much harder living conditions in these creeks.
Okay. Another question is, could the sick turtle be pushed into less comfortable water by healthier turtles?
I don't think so much as like necessarily pushed. I think these habitats are just, they are stressful, and there's also other factors out there as to why some turtles would be in better condition than others. It comes down to food availability and nutrient content and where there's better food and so on. So I don't think so much push. What would be interesting is to see, why do turtles not move when the conditions decline? And is that something that we might start to see in the future, especially if some of these habitats that we can see are starting to become, because of their variability, are potentially going to get increased to the point where just the conditions just aren't livable. And also, it's not just about the turtle at that point. They are obviously feeding in these areas, and therefore what happens to their food due to changing environmental conditions is also going to influence them as well. So it's certainly quite complex. We've also got reports of turtles being predated on by sharks, and certainly sharks are going to be targeting slower turtles, which are more likely to be in worse condition or more diseased. So lots of factors at play.
Okay. All right. So I have a question again for Annabelle by Sebastian Hofer. And it is, how is FP transferred across sea turtles, and how can other taxa act as vectors? And also, it's a two-part question. Also, does mortality increase in turtles with FP?
Yeah, so first of all, that yes, it does increase. So we don't see a lot of it here, but in countries, for instance, like in Florida, where there's a lot more people, so you're going to see more strandings of turtles, for instance. So a high proportion of strandings might well be there because of the FP disease, for instance. That's much harder for us to document here. And also, we really mainly, the studies that we do, the in-water studies in these coastal habitats, we're more likely just to encounter juveniles anyway, which is why we don't see it. However, these tagging programs and being able to visit these creeks is important, because like I said before, I've gone back into an area and I'm not being able to see turtles that I've tagged, where I would normally have a really good recapture rate, and they're most likely not there because they haven't survived the disease, because they're in quite poor shape, bad condition. So that's to answer that. In terms of transfer, it's a very contagious disease, and so just from being in proximity to each other, they could be passing on to each other, swimming in the same